ago because we were office mates. We were both postdocs at the Broad Institute. And there he worked in fungal genomics. Prior to that, he did his bachelor's and his PhD at the University of New Mexico. He also worked at the GGI. So he's been doing genomics for a long time. So, and also started with sort of fungal genomics, and then after his postdoc, went straight into industry, where now he's head of bioinformatics at Veritas Genetics. So this is co-founded by George Church, and it's one of the few genetic testing companies that does whole genome sequencing. So he's been involved in setting up all their pipelines there. I'll let him take it away.
Okay, great. Okay, thank you all for inviting me to speak. California is great. I love Santa Cruz. The name is great because I'm from New Mexico, so we have lowriders, and we always called Santa Cruz the patron saint of the lowriders. God bless the loward is what we used to say. You'll notice my talk is about genomics, so I was told that I am here to talk about also my career path, and it was circuitous. I'm glad I have a stick. It makes me look Gandalfian, because this is a Gandalf quote, not all who wander are lost, but I was pretty lost for a long time. And I'll tell you a little bit about that. So that's what I'm going to tell you about. Is I'm going to present this hopefully in a compelling way, since it was supposed to be for students. That me and the Human Genome Project actually had a similar starting point, not just that I'm human, but that we started in the same place. How we got here, I will tell you a little bit about sequencing. I'll tell you about the company's approach and why I joined them, and I'll tell you about what my role is, and hopefully give you some collected information about what I think would have helped me maybe not be so lost, and hopefully it helps you guys in your career path as well. All right, where did we begin? So I began in Santa Fe, New Mexico. That's where I was raised. Later on, I'll show you that my family's been in that area for like the last 10,000 years. Also, it's contentious a bit, but the Department of Energy, which heads up the sort of national labs, Lawrence Livermore, Lawrence Berkeley, Los Alamos National Labs. They were interested in radiation damage, and so they began to be interested in certain sections of the human genome. This led them to thinking, you know what, if we had the whole genome, we could do a lot more with this. So they were part of the group that decided that we needed a human genome project. It's thought that the Santa Fe meetings were kind of the coalescing point. There was actually one here in 1985 at UCSC that was sort of instrumental in bringing about the Human Genome Project. But it's the 1986 Santa Fe meetings, and later on throughout the late 80s into 1990 is when the Human Genome Project started. So we kind of have like a similar path. We started at the same point. I didn't know about genomics at the time, and not many people did. So here I'm going to present this 2 columns. This is what was kind of going on with the genome at the time. This is what kind of was going on with me. So I'm going to tell you a little bit about that. The genome, it had this sort of fracture split. It was like people thought it should be done way A, the old school way, and then a group said, you know what, we need to do it this new way, new, faster way. So it kind of split into this very, very bitter battle. But you know what? Maybe we should say that the competition, it's kind of an American, almost Trumpian thing to say, but the competition, it's about American competition, right? And that makes us better, and that makes us who we are, which hurts to say. But I would also like to say, here we are, Walter Gilbert, who won the Nobel Prize for sequencing technology, and who started the Biogen, one of the first biotechs, and Myriad, one of the worst biotechs there ever was. A field in which computer databases would be as essential as laboratory reagents would grow out of this. He was dang right. Okay, so now let's talk about, this is the same thing. This is what's happening in my career. I actually flunk out of New Mexico State University, and it's not even like a great school, right? Okay, I will tell you what, I mean, it's a good school. If anybody goes there, don't tell them I said that. But it's fine, but it's not like I flunked out of Harvard or MIT or anything like that. Why? Well, who's Hispanic in this room? Do you feel that your family encourages you to go to school in another state? Whose family encourages them to go into another state at all? Like, yeah, nobody does. Hispanic families would be considered in kind of like a way, we're considered enmeshed, and it's like you leave the family area and it's psychologically damaging to everybody involved. And so it's very hard to leave. So me sort of going even 300 miles away wasn't cool with my mom. I have a lot of family problems at this time. My mother is collapsing. She eventually does 3 months in a mental institution, treated for severe depression. I think that I'm going to give myself a break and say, right when I flunked out, she lost it. She had to be hospitalized several times. That had an effect on me. I think that's okay to say. And I'm going to be easy on myself and say that I couldn't deal with this. But maybe if I had some skills, it would have been okay. Also, you'll notice that a field in which computer databases were as important as radiation, it's not around yet. Computers are, who remembers the computer in the 1990s, in the early 90s? Did you like the computer in the 90s?
Oh, yeah.
I hated the computer. You were what?
I was a computer science major.
You were a computer science major. To me, it was very hard because I couldn't apply it very well. I was an engineering student at New Mexico State because it's a huge engineering school. It gets the most funding from NASA. At the time, it had the highest external NASA funding. We had to take a computer drafting course. Everybody's like, yeah, computer drafting, this is going to be great. So you plot your thing. You send your program to a printer. You don't get a GUI. It doesn't show you what you plotted. You send it to a printer. It says high-tech printer, and it takes 48 hours for it to come back to you, and you say, oh, I messed up, and it's not fast enough for me. I'm too much of a spaz. Who else thinks that they're too much of a spaz for this time frame? No? You guys are all too much of a spaz. This generation is all too spastic. You would never have made it, though. I'm kidding. You would have been fine. But I needed it to be faster, and it wasn't at this time. Okay. Where is this talk going? Here's what sequencing technology was at the time. I'm going to mix in some science along with it. Who knows this? Who knows how you sequence a genome laboratory-wise? A little bit? You do. You're not allowed to answer. I'm just kidding. Who else? You? Okay, right. So let's go over it. So you have a piece of genome, right? You have a piece of DNA, a long-stranded DNA from anything, right? You shatter it. You can also amplify these pieces, but we're not going to talk about that right now because we actually do some of this also. You break it into little pieces, then you repair those pieces and you sequence it from either end, right? Does this make sense? So you use this polymerase reaction, you sequence it from either end, and when you sequence it, this is what the Sanger Maxim Gilbert was at the time. You add radioactive DNA to it, and you only sequence you only add it to one base at a time. So you have one base for A, one lane for G, one lane for C, one lane for T, well, C and T, and then you use a gel. Do we all know what a gel is? Do we all? And then you use a gel, electrophoresis, and then it pulls this DNA down. And then what you have to do is you have to put an X-ray over this, right? And you let it sit there, and you retrieve the phosphorus signal. And I think I was gonna. Was there paired end sequencing at the time? Does anybody know from the old days? Does anybody? Is there anybody here from the olden days? From the old country? No, you may have been.
I wasn't in biology.
You were in biology. So I'm not sure if you could even do paired end.
Until later.
I don't think so. I don't think it was even around. But I'm going to use it. I'm cheap. I'm going to use this slide for a little time. Then you got some information. You don't even get you get like 50 to 100 nucleotides, I think, at this time. So it's very small. It's very slow. It takes a full day, I think, at least for that X-ray to develop, right? There's also some other things. Oh, I have a cutoff problem here. There's also some other things. They want to do this like fragment mapping, and it takes a really long time. I'm also going to tell you, what may have helped me through my psychological crises at the time. I would all suggest that we learn some coping skills. Coping skills include, which are self-care, which are, diet, exercise. If we too many beers, too many shots of tequila, which I love, it's going to be very hard for you to manage your mood. When you go into grad school, it's pretty intense. what we think now is that latest studies show that about half of grad students show signs of depression, of like real clinical depression. So self-care is going to be important. Exercise, eat right, develop some mental skills, and hopefully, you can get through this better than I got through this. Okay. Any questions so far? I think this is UCSC, right? We can sing songs anytime you want. We're like all cool with that, man.
It's drum circles here.
It's what?
Isn't it drum circles?
Drum circles? If we want to start a drum circle, let's just start doing it, man. Let's just do it. Right, and I also discussed the sort of family structures. Okay. So through the 90s, we kind of come back together. I recover. The genomic project recovers. We now go to a thing where we can like sequence a lot faster. We don't have to sequence by lanes. We can get longer reads. Genomics explodes. We have, Haemophilus influenza, fruit fly, worms. Anybody who's doing biology is starting to think, a genome could really help. This is in the day. This is sort of like in the cusp of when you could get a graduate degree doing just sequencing a gene. That's like you sequence a gene and you got a PhD. That's what it was. I know some people when I go back to school here in 1995 I went to a community college, which is fine. And then back to UNM in 1998, which is when I start at the Neurospora Genome Project. Neurospora crassa is a model fungus. It was the organism Beetle and Tatum used to do one gene, one enzyme. It was also what Barbara, help me out, McClintock used to find jumping genes at, so it's a sort of a well-known fungus. Two Nobel Prizes were granted using Neurospora crassa. Computers are now beginning to be useful, right? You can now see what it is that you were doing, right? And computers turn out to be, computation, which I think you all are doing. Who's not doing a computer thing? Is there lab type people? Okay, so most of you are doing computers, and now computers are very useful, and they're beginning to be integral in biology. Is, what's his name still here? Kent. Is Jim Kent still?
Yeah. Yes.
I kind of wanted to meet him. He's like a hero though, because he's taken over at this time. Right, okay. So with kind of sheer enthusiasm, I was allowed to get into Neurospora Genome Project. So here's the first thing. Always be enthusiastic, I would say, about it, and always say, if you say, do you know how to do this? What is your answer? Do you know how to do this thing? What is your answer? I can learn. I can learn and I'm willing to grow, because you will always do that in science. And if that's what you want to do, that science is your thing. And I'm still growing and learning. What else did I want to say? Okay, I think this is good. Questions so far? Any questions? No, stop me whenever you want. No worries. Okay, so Neurospora crassa, they wanted a genome. So they were dying for a genome. They tried to convince NIH to give them the money so they could set up their own genomic center. NIH said no, absolutely not. But we know these other people who are going to help you. So they contact people at the Whitehead. A man I eventually worked for, Bruce Beeren, who came up with the bacterial artificial chromosome at Caltech. And they said, why don't you do a genome for us? And it gets done for they wanted $50 million to get the genome done. And I think it really only costs $1 million. They're 30 megabases. They're not that big. They're kind of small. And so it was done, and they released it. My PI, she said, get the genome. I got the genome, and this is what it was. This was completely useless to me at the time, right? So this is sort of the first time I think, I have got to work on making this more useful to people. So this is sort of seeing this file made me think, we have to do better. And we're going to have to do better. Finally, I graduated in 2001. Lots of things are happening in the world. One of the most important things is that both the public and private genome projects, they decide that they are going to release their draft genome, which we can see a kind of a schematic here. Draft genomes are not together. We don't know what the chromosomes are. We kind of know. And so it kind of looks like this. It's kind of just a mishmash of stuff. And we don't know where one begins and when the other one is. And it may not be in the same order. So it's like a Picasso. I go to the Joint Genome Institute. Yeah. And I am putting together chromosomes back by back. At this time, we're doing fluorescence and slab gels, and then it switches to capillaries. So this is getting a lot faster. Instead of using radioactive dyes, we're using dye Terminator chemistry, and we'll say a little strand of a polymerase enzyme comes through, and it will add to single-stranded DNA. It will add a dyed Terminator, right? And so we know that this is a C cytosine, and it stops the reaction. And so these fragments come out sort of in different, come out size-wise. So you know that the start is here, the ending is up here. Does this make sense? We can also do it a lot more at time, and now we can get like 250 to 350 bases at a time. Also, we're doing paired end by now, so we can also say that, we can anchor fragments and say, we know that these fragments should be, 3 kb apart, and so we can start to use that information in order to put things together in bigger chunks. Also here, UCSC, they kind of create the method to make the golden, what they call the golden path. Right here, Jim Kent did that, David Hausler's group, and so they began to build the official reference, the official human genome. Yeah, it's kind of boring. All right, so at this time, we're also still using the back-by-back approach, which will be kind of, nobody will do this ever again. But the idea is that we would take these large, called bacterial artificial chromosomes, and put them together in overlapping sections. And here's like a map of what that looks like across the human genome. I worked on 5, 16, and 19 down here in order to put those together. 5 was brutal. It has large repetitive elements like this section, I think. If not, it's like a complete duplication of this section here across megabases. It was impossible to put together with the method we were using. So we had to like use, oh God, I can't remember the guy at UW's method in order to put it together. Long story short, nobody's going to do this again. But now we have a great reference to use. At the time, the cost for a human genome was still in the way up in the the 9 figures, right? It was approaching $1 billion. Does anybody not know what Moore's law is? Does anybody know what Moore's law is? Can somebody just raise their hand? I'm screwing with you. Moore's law is the rate at which CPU power increases, right? And so it's considered like the measure of how technology moves through time, how fast we increase technology. And you'll see that the price of genome sequencing is dropping faster than Moore's law. Okay, but we're still up here at $100 million. So at this time, we've done enough finishing. We're going to move on to other things. Department of Energy has interests elsewhere. I get put onto leading, which was crazy because I didn't even have a PhD. I didn't even have a master's at the time. I worked for somebody who was absolutely nuts and decided like, yeah, he can do it. That's fine. Let him do it. Who knows Dan Roxar? Yeah. Man, it was crazy though. Like, I don't know how he gave me this kind of power, but it was great. Because now I have a first author paper on the cover of Nature Biotechnology. This is my project right here in 2004. And you can see we're studying a genome of a white rot fungus. And so what these fungi do is that they completely degrade all parts of wood. And this is it growing on a little section of wood. This is a scanning electron microscope. How it does that is kind of interesting. It uses enzymes that are high molecular weight, and they create like peroxide and oxides, and they just blast the crud out of wood, which breaks down the lignin so that they can get out the cellulose. Here's my first kind of interest in analysis, is that we started to notice that if we compare the genome of white rot fungus with other sequenced fungi, we have an expansion of genes with this cytochrome P450 domain. And this is the enzymes that are responsible for this degradation. So we now develop the hypothesis, or sort of, we continue to expand on the hypothesis that a gene expansion in a microbe indicates that they have some phenotypic ability, right? Any questions about that? phenotype. It can do something, and it has something to do. They're also sort of structured into particular regions of the genome. We continue this with, this is my first publication of my graduate program, so I go from Lawrence Livermore National Labs back to New Mexico to Los Alamos National Labs and find out that the Trichoderma, which is a fungus that is studied for its ability to break down cellulose, that those enzymes are arranged in certain areas. They're concentrated in certain regions of the genome, and they co-express together, as you can see. So xylanase, cellulase, manocytase, here's chitinases, here's a bunch of other cellulases. So this is, and the interest here for the Department of Energy for both of these projects is trying to take corn, just unusable corn stover, so this is like the cob, this is the stalk, the leaves, and trying to break it down and make it into ethanol, right? That's their goal, right? And this is actually a Bush era goal. When Obama comes around, they abandon it because this was a Bush era goal. So this is kind of sad. It's abandoned now, based on political affiliations. Right. Any questions about this? Yeah, go ahead.
What exactly are the arrows showing?
Yeah, the arrows are genes. These are genes, and they sit in the genome, right? And here are, if you're blue, you are a carbohydrate active enzyme that's not necessarily related to cellulose production, but the green ones are. The green ones are genes in the genome that break down cellulose, and this whole region sort of co-expresses, almost like a pretend operon. Does that make sense? Please go ahead.
Those are just like the direction of transcription.
They are, yeah. They're the direction of transcription, though. And that was important for to see if it would co-express, like if you could figure out where the promoter was based on just how things were facing. Okay, great questions. Thank you. Thank you for helping me with that. Okay. Let's see. So here's where I began scientifically to be very interested in, this is kind of strange, but how the genome is organized, how it's kind of laid out. So at about the same time, I'm sort of switching. This is 2007. I'm in graduate school. I was a Department of Energy fellow, and I went to the Nobel laureates meeting in Lindau. Has anybody heard of that? No. So every year in Germany, they have like a different sort of Nobel Prize section, and they invite any laureate who wants to come to meet with students. So I get to go. And I was at the 2007 meeting, and this was really a lot of fun. And I talked to this is Richard Roberts. He discovered the intron. We all know what that is. He got the Nobel. This is Craig Mello. He discovered he helped discover RNAi, which was sort of a big thing for a while. I can't remember what he did. He discovered nitrous oxide signaling. So Viagra, thanks to him, we have Viagra. He's not proud of that. What they say is the most important prize in Nobel medicine coming for the future will be the $1,000 genome. So this is like, I was like, wow, that would be great to be a part of that. So I'm just sort of like foreshadowing a little bit here what's going to happen. I think in grad school, I started to notice that a lot of us, I was sort of, I'm going to get back to touchy-feely stuff. So let's all hold hands and sing Kumbaya, Santa Cruz style. I'm just kidding. Who has heard of imposter syndrome? Yeah, okay, good. So at the time, it wasn't sort of well known. But it began to see that, a lot of people were feeling that anything that they did successful was by luck, that you were a fraud, and that you will soon be discovered. And this causes hell of a lot of anxiety, right? This is very difficult. So if you do feel this, you kind of need to, this is almost like cognitive behavioral therapy, is that when you have a negative thought, you talk the negative thought away, right? When you have that feeling, you sort of like think about what could be the reality behind that feeling, and that's kind of a cognitive behavioral therapy. You get lucky, you're already here, right? somebody has already seen that you're okay. But don't try and rely on external validation. Like, you want to be here, you want to have something to do, right? You've been discovered as smart, right? You're doing the best job you can. And every time you have a success, you're getting better at what you do. If you think that you're a fraud, it might help to realize that there are ways to sort of think about yourself as somebody who should be here and feel and help to make yourself feel included. I think that with programs like this, I think people are starting to feel a lot more included, but I think at the time we didn't feel super good at that. But I think we're all doing better. And also, of course, think about a situation and what your reaction to the stress might look like for yourself. How would you like to handle this, right? Okay. So we're here. I'm going to switch back to the genomics thing. We're here in 2007. You'll notice this wicked drop here, right? So what happens is that Illumina develops a different type of, well, I don't know if they developed it, but sort of they popularize a different type of sequencing. Yeah, right? Call it sort of sequencing by synthesis. The difference is now we're looking at the signal when a base is added. Instead of like sort of dying the signal and then trying to figure out what it was after, what you do is you sort of lay the DNA down on a slide, and it forms these sort of like these bridges, which then open up to single-stranded, and a polymerase enzyme comes along. But now what we're detecting is we're detecting each individual signal as a base is added by the polymerase enzyme. And that base also has a terminator, a reversible terminator, so you don't add too many at once, which was solving a problem by a previous company in which you could add. If you had like a homopolymer string of A's, you would get this giant signal and you couldn't tell exactly how many were there. But now this, with reversible terminator, you could grow and see. Questions? Does that make sense? All right, good. All right, okay. So at this time, I graduate. It's 2010. I graduate, sort of thinking about, what to do next with my career. I begin to think, if I'm going to have to fund myself, I think that NIH has the most money, so I'm going to try and go after fungal diseases. Fungal diseases are actually really, really damaging. We'll say in 1 year, 500,000 people in Africa will die of cryptococcus, right? Cryptococcus is a fungal lung infection, and it's super common in sub-Saharan Africa, where it was largely contracted by people with HIV who are HIV positive, but then it's begun to spread to other people. It's kind of controversial. It's like if you take cryptococcus and add it to tuberculosis, 1 million people die every year of two microbial diseases. So this seems like it's kind of important. So I joined this group. Kind of important. I joined the Broad Institute, where we were office mates, and began to study. Well, let's not worry about that. And I studied, instead of like studying cryptococcus, which is actually kind of hard to do because it's deadly. It will kill you. They put me on this project for dermatophytes. It's athlete's foot, but it's a pathogenic fungus still. $500 million a year are spent treating athlete's foot. It causes a lot of stress for older people, and we have an aging population. It could be a problem. It is a problem for people with diabetes. They have like very bad infections that don't heal, and this makes it worse. What I discover is that here are a group of genes, and dermatophytes, which are the red phylogenetic branches, and the other branches in other colors are other types of fungi from the Aspergillus. So that's like bread mold, Candida. That's like thrush or stomach yeast. This is mushrooms here. The red branches are enriched with a particular group of genes that contain these red blocks here. These red blocks here are domains that probably originated in bacteria hundreds of millions of years ago, called the lyse-M domain. And what they do in eukaryotes, and this has been shown in plant pathogens, is that they disguise the fungus from the host immune system, right? So you can't get rid of, I can't get rid of, we can't get rid of athlete's foot. And we're proposing this as the hypothesis that the expression of these genes means that your immune system can't see it very well, right? And so it becomes sort of quiet and disguises itself, like my voice right here. Like I'm emoting the voice. Questions about this? Does this make sense? Right? Okay. So the funding for this dries up. It goes down the tubes. Let's see. And actually, I studied Candida for a while, but I forgot to put that in, what I found with Candida. But that's fine. I can tell about it if anybody wants to know. So let's look at this. So it's 4 years. The NIH has this wicked drop in funding. We have 2 grants. The group I was with has 2. It's 4 years into a postdoc anyway, so it's time for me to do something else. But our group had funding from NIAID. So that's the National Allergy and Infectious Diseases group, the section at NIH, and they're like, I can't remember how many millions they are, but it's pretty big. We have like a giant grant from them, and we reapply and we get it. We have like one of the best awards ever. And but it gets like everybody just that year, that funding cycle, everybody just got a 40% cut right off the top. So right away we got a 40% cut. The Broad Institute's biggest grant was from the I think it was. Is it the Genomics Institute? Is the NHGRI? I don't know, but it funds most of it, and it's like hundreds of millions of dollars. It's a massive grant. It gets cut by 50% too. So I'm in a group that's 100 people. It gets down to like 10. Like we did down to 10 within a year just because of budget cuts. So I'm thinking about this. I also tried for some postdoc grants, and I'm not as good a grant writer, and I can't kind of feel it like Angela could. She was so good at it though, and I was never able to do it. I would have to say, who has like some sort of like, who's a graduate student? Are there graduate students here? Do you guys have like a graduate student funding external? Raise your hand if you do. Higher. Come on. I'm just kidding with you. Okay, yes, this is very good. Is it NIH or is it NSF? NSF, all right, okay. I would have to say that NIH is really tricky if you're not doing non-human, they're not as good. And I didn't kind of know that going in. There is a grant that I wanted to get that I thought about getting. It's called a Kangaroo. It's a K99 R01. So it funds the end of your postdoc and the beginning of your independent R01 research career. For fungal diseases per funding cycle, how many do you think they give away? There's 3 funding cycles a year. How many do you think they give away? 2. They give away 2 a year. And I was like, I think I can be competitive, but I can never get, I never did it. It was not successful. But then I found out if I did get to R1 and I did study fungal diseases at NIH, how many grants per funding cycle do you think they give away? Any guesses?
Less than 10.
1. It's 1, yeah, for fungal diseases. It's only the one institute. And I tried to go through other institutes, but they say if you're studying the organism, they shift you right back to NIAID. And I talked. So if you do have problems, the NIH trick is talk to the program manager and see what they're thinking, right? So the lesson there, and I think it's this way at NSF, it's like you submit a grant, a scientific review board reviews it, right? They look at it and they score it, right? They give that score to the program manager. That is not the decision about who gets it, right? That is only an advisement to who runs that program. So you have to know that program manager very well, and you have to do exactly what they think is important, right? You want to tailor your grant to meet their mission because they're the ones who actually decide. They will take that scientific score and they will take it under consideration, but the final judgment is theirs. They can take a lower one if it meets the NIH mission, and that's what their job is. They need to have research that goes toward what the NIH mission is, right? Any questions about that? Right? Okay, sounds good. Right, oh, here is a graph from, I think it's 2015, 2012. So this is 2012, 86,000 biology PhD students. There are 30% of them will do, 30% will do more than one postdoc. And I wasn't going to do that. You have 29,000 people who are in tenure track, 7,000 in government researchers, and 20% more are in non-tenure tracks. So what we see is that it's something like only 8% of graduates actually, at this time, only 8% of people who have a PhD will become tenure track professors. You know, and I was told this, and I thought, you know, I don't care. I still want to be a professor because I love research, though. It's like what I do. But there was some sort of soul searching. I could never get a grant. It might have been better if I tried to go for some genomic evolution type of focus. It might have been better if I'd done that. So maybe if I had sold it, it would have been better. I'm also not that kind of well organized. So I'm sitting there and I'm doing the all your kind of you have to do these plans in order for your tenure track faculty position application. You have to do plans of what your teaching statement is. I'm just I can't do it anymore. So I walk into we had a lunch together, the four of us. We were 4 postdocs. We would hang out together. I had a lunch and I just, I said, I'm out. I'm not going to do it. And a friend of ours said, I was talking to a recruiter and there's this startup that is desperately looking for a bioinformaticist. I was like, okay, fine, I'll talk to them. And I think it works out pretty good. This company is called Veritas Genetics. And since then, it's been around for 4 years. I call myself employee number 3, but it was really number 6, but 3 people in front of me left. So I've moved myself up in the ranking. I'm employee number 3 now. We start getting 50 smartest companies by the MIT Technology Review. We always get it. We get it in 2017. We'll get it again in 2018. I will tell you that it's being smart. We're ranked above IBM. If you can believe that. We're ranked above IBM in this review. Also, our CEO knows the editor, and they play golf together. So also, if you're going into industry, which I'm never going to be, if you want to be a CEO, your golf game is really important. So don't forget that. That's why Trump plays golf all the time, though. It's that's how you make stuff. But I still think, fine, we deserve it. I'm not going to imposter syndrome us out of that, though. Disruptor, everybody wants to be a disruptor today. If you're at a startup, you're yeah, we're disrupting, we're going to change the world. So we finally got that. Apparently, we're the world's most innovative companies, but we'll see. So now we're down here. I started in 2014. Genome, because of technological innovations, it's really cheap. We don't start with the whole genome. Rate my setup, fam. This is what we have today. This is kind of out of order. This is what we have today in order to get this done. We have 3 NovaSeqs. This is their big new one. This is what they think. This is what the company thinks it can drive down the cost down to $100 a genome. That's what they say. We'll see. It can do 48 genomes something like in 2 days. It's something like that. We have 2 high-seq X's. We actually bought 10. And also we have all of these in China too. We have a China office too. So they have all of this too. And they're like madly doing Han Chinese. We also do some panels, and I'll talk a little bit about those. And so we have these smaller ones. So we have anything. And so we'll sell sequencing to anybody who wants it. And we'll do whatever you want too. We'll do development, anything you ask us. We'll do it. I don't know why I put this slide here. But let's see. All right, and so here, and one of the reasons why it costs so little is we're actually just resequencing everybody. So we're not doing like this de novo sequencing and assembly. We're not doing unknown. What we're doing is that we're taking all of those sequences. Does this make sense to everybody? So each one of these is a read of DNA that comes off of a raw machine. It is aligned to the UCSC reference. So I still use the UCSC HG19 reference. And here is a variant at this position. This is actually my genome. And this variant here puts me at risk for opioid dependencies. So I'm going to never going to take any type of opioid because I'm going to wind up on the street corner. It's not going to be pretty. So this is something it's good to know. We don't start there. We actually start with breast cancer. Breast cancer, at the time, it's kind of controversial to have a test for breast cancer. But here's why we start here is because it's important because if you have a pathogenic mutation, I'll talk a little bit about what that is. In fact, here is a description. Here is, let's see, let me get oriented here. So here is the domain in the BRCA1 gene. Does this make sense to everybody? So this is a representation of the BRCA1 gene. Here is the BRCT domain. So this is a 2-part domain. And here's like the protein structure of that. Right? Does this make sense to everybody? Here's when the protein is folded. This is like a helix here. These are helix here. I don't see any depleted sheets. And it fits to DNA because it's a DNA repair enzyme. That's what it does. DNA repair mechanism, part of DNA repair. Here we have an insertion of a C here, and it causes a stop codon, right? So it causes a frame shift mutation. Do we know what that is? Who does not know what a frame shift mutation is? No? Okay, good. So what happens is, you have triplet bases. If you mess that up, if you insert something in the way, it screws up how those triplets are read downstream. This causes this domain to become truncated, and it affects its function. This will cause breast cancer. If you have a pathogenic mutation in it, it increases your risk of breast cancer by 40 to 80%. And it's also sort of like the older you get, the worse, the higher the risk is. And so to know this, it's actually somewhat preventable. You can have a hysterectomy, you can have a mammectomy, and greatly reduce your risk of death. This is kind of a strange study, and I don't know what to really think about it. It's somewhat controversial. I don't know even why I'm talking about it. It's an Israeli study, and it says that the younger you are, actually, the worse this risk is. We might say that it's really because of better detection is really probably what's happening. But you will hear some people say that there's something happening, and it's like radiation and whatnot, and dry diet and stuff like that. So I'm sorry it also to be skeptical.
We actually have a resident expert here. Melissa Klein is one of our Genomics Institute research staff scientists who's working on this very question. Yes.
Tell me about it.
Well, with 3 people here.
Great. What are you guys looking at?
Well, we're pulling together evidence that describes whether a variant is likely to be pathogenic.
Fantastic.
And
That's
we're pulling together clinical reports from Clinvar population frequencies. We're working on getting other types of evidence from testing labs, such as how many times they have seen the variant in a family that's got a lot of kids.
Yeah, right. So proband. Yeah, provide information. So that's great. That's great. That's excellent. That's an excellent. And so we're doing the same thing almost, because I do something very similar. Right. Okay. And this is the sort of company story. Right now, the patent was at this time. The patent was held by a company called Myriad. I had mentioned them before. They're charging so much, and it's so critical. If you're part of an at-risk group like Ashkenazi Jewish or French Canadian descent, there were bottlenecks that concentrated pathogenic mutations in those populations, and so you could be at risk for this. It's something like 25 or 30% of people with breast cancer have a mutation. So it's not like whatever it is, the Bayesian probability isn't quite as high as you might think, but and you would still have a $200 to $500 copay depending on your insurance company even then. Also, the share of healthcare spending is going up. The CEO is Balkan, so he's like always kind of predicting this global balkanization that might happen. Maybe he's right. I don't know. It's hard to say. And so we launched in 2015, $199. So it's cheaper than you would pay for the copay. Plus, we will give one away. So we will do 1 for 1. We will give one away to anybody who gets it, and we'll give one away to somebody in need. And so we work with. It's not common, but it's like several different foundations in order to provide tests for people, right? And so this sort of establishes our mission is that we're also not going to hold on to, we're going to share the data as much as we can with whoever we can. Also, shortly after this, we announced that we would now sell data. We will now sell a genome directly to the public. We still have to have your doctor's permission for $1,000. So for $999, we would sell you a genome. We also do research services, but that costs and we will give you any type of raw data you want, but it costs $1,500 base.
Do you mean whole genome in that you've mapped the reads to a reference?
This is correct. We have mapped the reads to a reference, though. We'll also sequence anything. Somebody's now going to try and sequence goat. Sorry? For $1,500. We'll charge you extra for raw data, but we'll also interpret. And that's what I'll be talking about. Right? We'll also tell you what your risks are. Right. Now we want everybody to get a genome. And so we're going to try and work hard for that, though. We're going to go to maybe other subscription models. So it's like you pay a little bit more so that we keep telling you what the new risks are when we get more and more and more. Also, we're working with, since we are a George Church Foundation, sometimes I look at you and it's like George Church is sitting right there. Because you look just like him. He has a giant beard like that. And that's why I kind of stare at you. It's like, oh. The man is here. I am not worthy.
He's imitating Darwin.
He is, right? He was trying to get it white. His daughter has a beard oil company, and so I use her beard oil for my mustache. Yeah, true story. That's like her thing. There's also another George Church startup. He has 35 of us out there, and they want to do do blockchain genomes, right? They want to you get a coin, and so somehow when it goes public, we will actually get the money to sequence it, right? And it will somehow work. It's really not clear to me, but you're going to blockchain the genome. It also is a good way to identify it too. So it's we could all run around. We could all balkanize. You could have your own currency. You could have your own genome, and it would all be somewhat safe, right? So that's a possibility for the future. That's thing. So this is what I do. I run the clinical pipeline. This is sort of a mishmash of stuff that you will see. The first half, this is all sort of standard processing. This is what the machine is, right? So here's a HiSeq X. You get these fast queues out, like you were saying. It's the raw data. We then align it with BWA-MEM, which is just what everybody does. I actually use 2 variant callers in order to get my my sensitivity higher than most people. So I use this sort of combined technique. I get more false positives. So False positives are things that I say as a variant, but maybe it's not really a variant, because I don't want to miss anything. We're going to filter it out later with other methods. And then also, I will talk a little bit about this, because this is what I do, is run this interpretation step here. So right, we're going to, here's a variant, and I talked a little about this. All right, that's fine. Here's a record of a variant. Who's have seen a VCF file before? Yeah, not too many people. So it's there's also some people like barely raising their hands, but that's fine. You know, this is UCSC. Chromosome position. This is what the reference was. This is what the alternate is. So this is what it was replaced by. This is a quality score. This is whether it passes a filter. There's a bunch of other stuff here, and it tells you that this is also here. Is the important part. This is also tells you it's heterozygous, though. So we all know what who does not know what heterozygous is? All right, we all know what heterozygous is. That means we have 2 copies of every nucleotide, and one of them has changed, right? And now we want to know, for that variant, how does it impact our health, right? This has been a goal of mine, is to be able to look at this data and be able to make sense of it. How do we know? And how do I weed through the number of variants? There are like depending on how I run the VCF filtering, it could be between 3 and 5 million variants per person, right? That's a lot to look through. And so here's a brief statement of how I look at it, since we kind of know a little bit more. I'm going to run it through 3 steps. I'm going to first try and figure out its biological function, right? I am then going to try and figure out how common it is in the population, and then I am going to figure out if it's been seen before by another clinical testing lab, right? This is performed largely through the script Anavar, but I'm going to make my own soon. All right, here's a disease. Let's all play together. Let's all try. Let's all play. This is a fun little thing. Let's all annotate this variant and curate it and see, is it pathogenic or not? And let's look at the evidence, okay? Right. I'm going to describe what she just kind of just said, determining allele frequency. So how common is it in the population? So let's say we're dealing with frogs. Their color is determined by a gene, right? And we're going to calculate how frequent that is in the population. So this is, I think, these are heterozygous, these are homozygous. So these, if this were a disease, these people would be affected, and maybe they would die early or something like that, right? So we're going to see how common this is. The most common disease, by the way, the most, I think, is still delta F508. This is a CFTR mutation that causes cystic fibrosis. It causes a loss of function mutation to this CFTR. It's a chloride ion channel membrane protein. You know, interestingly enough, and I don't know if this is really what people think, it's fairly common because as a heterozygous, and it's fairly common in African populations, as a heterozygous, it would actually prevent you from becoming too dehydrated due to dysentery, which is fairly common in the third world. Does anybody know that that's not the case? Or has anybody thought that that's not the case? Is that okay? Is that kosher? Right? And if you're homozygous, you have cystic fibrosis, and your lungs fill up with fluid, and you die at like really an early age. There's treatments actually for delta F508 and a few other mutations, but they are hard to deal with. Here's what I just described: is this delta F508 mutation interrupts the contact between the transmembrane domain and this activation domain here, and that's what it does. The most common one. Where did I get these out of order? Okay, right. So that's its functional impact, right? So we talked about frequency. Now we're going to talk about function, right? This is its function. It actually does impact the protein in a particular way. Also, we're just going to Google it with Clinvar. Clinvar is the standard database that everybody who's ever seen a pathogenic or non-pathogenic mutation will put it into this NIH database, right? So Clinvar is where we look and we see what other people think about it. So we're kind of polling for it. Okay, so let's take a look And so this just says what we do. So this is, I take like something like 5 million variants. I run it through these three filters, and I get down to something like 10 or 20 per person that might be clinically relevant. From here, we do, manual curation. So let's look at the evidence for this mutation, right? Its frequency in EXACT, so there were these large genome projects, EXACT being one of the first collected at the Broad Institute. It's a bunch of exomes so that we can, there's something like 100,000, I think is what it is, roughly 100,000 individual exomes that's in this database. And so we scan it for frequency. Its frequency is low. So if CFTR is disease causing, it's at 5%. And this is under. So this is under or over 5%, right? It's under. That's right. It's under. SIFT and polyphen are two tools that estimate if it actually has a impact on the protein folding, right? One of them, SIFT, says deleterious. The other, polyphen, this guy, the guy who writes polyphen is on our board of our scientific advisory board, says possibly damaging. And we know it's a missense variant, so it causes a change in the amino acid, right? So it alters the amino acid. And then amino acid altering is somewhere here, but I can't find it. Can anybody see it? It says, all right, it's in red. It's an alanine to valine change at 114. So it's kind of early in the protein, to tell you the truth. It's kind of early. Who thinks it's pathogenic? Raise your hand. Who thinks it's not pathogenic? Raise your hand. Yes? No? Who doesn't care? The pathogenics win. So we report this as Homocystinuria, and it goes in a carrier. So this person is a carrier of Homocystinuria. This is not me. I actually don't have anything that's kind of this crazy. And I think this is an Ashkenazi Jewish variant that's common in Ashkenazi Jews. And it causes cysteine metabolism. It can cause thin bones, scoliosis, sort of like glass-like syndrome. Any questions so far? Right. Yeah, please.
So when you say that you take the DCF, do you go per record, like per variant, or do you see a combination of variants to do this kind of inference?
That's a good question. Right, so right now, we're looking just at one variant at a time. I do have ways in order to sort of look at multiple variants, but I think we're kind of just at the beginning right now of looking at multiple variants. There are ways with GWAS, but it's sort of like, it's not, I'll talk a little bit about that, and oh no. And we're still at kind of the single variant, one disease, one phenotype. I think, we're quickly seeing that those have been exhausted. We now need to start looking at multiple variants, multiple effects, and multiple things, and I'm really trying to drive in that direction. So that was an excellent question. Thank you very much. Excellent question. All right, any questions about this? No. Okay. That point at which we get down to those 10s or 20 variants costs us the most amount of money, right? We have 100 curators. We have physicians. We have like genetic counselors who are all scanning literature all the time, right? We have people with medical degrees who are just doing literature searches in order to figure out if what we've found is actually something that causes disease. We think that with deep learning that we could make some gains in this area. We could bring down costs. Since we're a low-cost company, everything is cost-driven, and this is something that you'll find in a lot of industry. That's kind of the one difference, is that you want it to be costing like minimally. There's sort of like a interplay between clinical teams and research teams. And clinical is very conservative, though. You don't want to go off and say anything that somebody might come back and say that that's not right. So they're sort of recalcitrant to new methods, but we're working on it. Also, if we had full text publications, we could probably do a lot better learning, I think. That would be great to have. Question, yes?
Do you use the publications track on the Genome browser?
We use other things. We use proprietary things like HGMD
Well, you want
and
focus
stuff
on
like that.
publications. All publications have been mapped to the genome.
Okay, great. Thank you.
So if you're looking at a section of interest, you can see what publications belong to that section.
Okay,
Yeah.
we'll look at that. Thank you. Thank you. Thank you for that tip very much. Everything? Yes.
How do you weight different external databases differently? Like HGMD maybe has some bizarre mutations in it compared to Clinvar or something. How do you guys handle that?
Yeah, so that's in weighting. So the question is, in case somebody didn't hear it, do we weight different databases differently? And I would, and I will work with that as a weighting, because HGMD does have some strange things in it, though. It's not clean. So HGMD is the Human Genome Mutation Database, and they claim they pay a lot of money. We pay like almost $100,000 a year for it, for 5 people to look at it at once and to have like the download. They claim it's like heavily curated, but what I really think they did is they just said this variant is in this gene, and this gene was found to have some association with disease. So it's really hard to trust. The amount of overlap between, so Clinvar is like what most people use, it's freely available, and HGMD is remarkably small. And I don't know if I trust it. Do you guys use HGMD? Do you guys look in HGMD? You guys probably get it free. It might not be worthwhile though. It may not even be worthwhile though. It's hard to use. Yes, sure.
So if you are saying that your variant color is allowing false positives, and then you are using public databases for the association, and how do you do this testing? Like how do you do?
Yeah, we'll resequence it to confirm it.
Okay.
If we think it's close to. There's like sometimes there's like a gray area where a variant, it's probably like a quality mapping score of 100, which means that the risk that it's a Fred score, it's the risk that it's an error is for 100 is like what? It's 1 in 10,000, something like that. So if it's near that, we'll Sanger sequence it. So we'll do like old school sequencing and see if we think it's still a variant though. No. Just that we'll make special custom primers, and then it will just PCR that one section, and then we'll sequence that fragment, and we'll do that. We actually get GenomeS to do that, though we don't do that in house.
And for now, you don't use any statistical model to know how much you're losing for that.
How much?
How many times you have to do this?
Right. So is it worth it to even do this? Yeah, I'm looking at that. So I am looking at that, and then to see if I can back off. I'm also looking at using deep variant, which has like a higher accuracy rate. Deep variant is sort of the new. We were talking about that last night.
You've been running deep variant.
Oh yeah, you've been running deep variant. It's slow, isn't it?
Yeah, it's not. There are ways to make it faster.
Okay, yeah, let's talk. I had an undergrad, so I still take students. I have had like 8 undergrad students that I work with. We get from local universities, so we still do lots. Okay, am I running out of time? Did I run out of time?
Close. Yeah, we're gonna move into the Q&A pretty soon.
Okay. Darn. Also, we can tell ancestry. This is me. This is my ancestry. You'll notice that my mitochondrial DNA, which you only get from your mother, is B2A2, so I'm really proud of this because it originates in southwestern US. In New Mexico, actually, is the epicenter 10,000 years ago, so that's fun. I'm also, broadly, I have Native American, about 16%. This is autosomal. I'm also a little bit sub-Saharan Africa, so shout out to my brothers. Sorry, that may have been not quite politically correct. And also Ashkenazi Jew, so I'm everything. Like Mazel Tov. I can do everything. The only thing I don't light up is the Middle East and Australia and Asia, but still even large parts of Asia. We also tell you other things like whether you have, this is actually mine, star 1, star 2. So this means that for a particular drug metabolizing enzyme that's in my liver, I should probably watch what kind of serotonin reuptake inhibitors I take. I shouldn't take certain types though. So this is something that actually is useful for me. We also do GWAS. I don't know where I'm going with this. We also did US. If somebody wants to talk about that, we can. Where are we going with this data? We're going to use the GA4GH Global Alliance. We're part of that. We have what other people have had the idea of. We bought a company. They have this platform called Arvados. And so when you sequence with us, and you will allow us to, and you consent us to, we'll have a method for like having researchers come in and use that data. And so it's unmarked, but we can share some phenotypic data if you let us, right? Also, we also sequence for the guy who was responsible for the ALS Ice Bucket Challenge. He's kind of heavily responsible. He's a researcher at UMass Worcester, and so we do his sequencing. So we do lots of research sequencing. In fact, that's probably mostly what we do. It's almost like the direct-to-consumer version. It's not really direct-to-consumer. Strike that from the videotape right now. Because the FDA will, like, I can hear the FDA helicopters right now. You still have to have your doctor, they have dawn rates. It's dramatic. You still have to have your doctor's permission, but it almost just gave us really good press in order to do that. So when a researcher comes in, we work with them to try and get the consent right. The consent is the thing that you say, I allow this researcher to use my data, and we will try to say, and it can be used for other research purposes as well. And we're trying to help people do that. I'm sure that you're working with that also, trying to get that consent right. And you'll be able to use it within the Arvados environment. And we have tools for everybody to use the data that you share, and for all researchers to be able to share within our Arvados cluster, that you can write your own code, write your own Dockers, do the whole thing within Arvados. You can actually get a free account. Consent remains difficult. Also, we just kicked it off. I guess I'm able to say this. It's kind of risky here. NIH's Million Genomes project, we just joined a consortium, and so we will try to convince NIH, again, here I am, all over again, writing a grant for NIH. It never ends, to also sequence 1 million genomes for the NIH's Million Genomes All of Us project, though, which I recommend everybody join. Because I think I don't know, I think you get like a low cost or a free genome out of the thing if you go through all this testing that it has it. Okay, career do I still have time to talk?
You can, I will just say that anybody who needs to leave, we understand if you need to leave. If you want to stick around, we have the room. We're fine. You can keep going, Diego. We'll plan to break around, say, oh, actually, you're doing good. You've got until 12:30. Excuse me.
Okay, I'll just keep jabbering, blah, blah, blah.
If you need to go past 12:30, you've got a little time.
Okay, I don't think I have that much. All right, here's a career comparison chart. So I did 6 years in government. I did, time, sounds like I was in prison. I did time, man, in academics. And so now I'm at an industry startup. And so I just wanted to give everybody kind of what it's like and what the differences are with this chart. If you want me to add a column or if you have a question, please ask. Feel free to ask, no problem. Is there bullshit in these managerial structures? There is, but it's just sort of different types, right? You have your political bullshit in academia. In government, you have a lot of weird external things, like an administration comes in and this director wants this push. And then that changes and this director wants that push. And then suddenly you thought you were doing fungal genomics. Like everybody I knew who was doing fungal genomics for biofuels, they're all now doing algal biofuels, right? And it's not what they came there for. It pays fairly well, so you're kind of like in a weird place. Like you can't really leave as well. Academics has weird internal politics that kind of runs amok at times, and people can get into huge fights. And yeah, industry, I'm at a startup, and so it's like slightly different than normal industries. There's still some politics, but everybody's pretty much just working so much that it's just, let's just get it done, right? So I kind of like the environment slightly better. Do you have academic flexibility? Again, it's administration dependent, right? It depends on what the administration wants, and it depends on if you meet that goal. Academics, you still have to convince somebody at some funding agency that you should do that or get like a Pew Fellowship or something like that. Academics is probably, you're going to hear this in it, I would agree, it's the most restrictive. And if it's going to make us money, or if you have the time elsewhere, you can do it. And also, with the sort of time flexibility, there's a lot of people, we all have like side hustles, we all have side gigs, but you guys have side hustles and side gigs too. So for example, I have, my own startup, which we make technological devices in a weird way, and I can show you the movie that I made for it. Deadlines are something that like really stressed me out. Who gets stressed by deadlines? Like everybody. If you're a grad student, you're like, so government, like the deadlines are kind of really strange and flexible, and I see a lot of people sort of able to kind of skip through deadlines and be able to like keep their jobs somehow, and I'm not really sure how they're doing this. And so it's kind of strange as to how it works. It's like not a real deadline. It's like a congressional deadline. It's like so screwed up. Academics has these hard grant deadlines, and that's the hardest deadline there is. Like this has to be here by noon. That is absolute. And then you have like testing and stuff like that. So you have some hard deadlines that are hard to get around. Industry has like the biggest bullshit deadlines. They have like the most thing like, we have to do this by this then. And nobody has a good way of measuring how long it should do anything, especially when you're dealing with technology. It's like, so it tends to be the most inflated, I think. But it's actually, I think we'd have to say it's the easiest to manage. This I'm trying to get at also the changes in government were so difficult for a lot of people who are doing research. And when do things change? Like when grant, you see new people change in academia. When administrations come in government, there's like sort of a whole new group of people that come with it. In industry, at least at a startup, we're constantly chasing. We may change what we do because somebody will give us money for it, and that's just the way it is. No, that's just you're sort of chasing a buck. Right. The one thing that really hit me hard about academics is how much like sort of upfront organization I needed, and it was like almost too much for me. So I think would have to say, if anybody disagrees with any of these, they can chime in. I don't care. We can talk about it. We can fight about it later. It's okay. Government, you have a weird organizational thing in that typically if you're at like a national lab or something, you have a security department, and these people are like, oh man. At Los Alamos, they were called safety and security. And I'm like, does anybody get it that that's the SS? Like, does nobody got this joke? Like that they're called the SS directly? Come on. So you have to get through the security, and man, it can be horrible. In fact, I kind of just started to ignore it, and nobody said anything. So I hope I could still be, it could still be coming to get me. Sort of at an industry startup, you don't, because it's kind of incorporated into the amount of time that they pay you for your time. So they're not going to pay you too much. Like if we have to do organization, something's not getting done. It's like we just need to get it done. So like stop organizing and go get it done. So I think it's kind of the least. How much you have to hustle to get stuff done? I don't even know why I put this in here. It doesn't make sense anymore. It's kind of like what I was thinking of. I think you have to really hustle hard everywhere you are, but how much you're expected to hustle externally, how much pressure you're put under is kind of variable. And so it would be like sort of less, I think more like academics. You have kind of the most of that. And it depends on your role in a startup. In a startup, it's like we're all doing so many different things. So it's like I was head of IT at the same time I was head of bioinformatics. At the same time, it was like I was the only person who knew computers, so I had to I did networking infrastructure. I did everything. Everything was me. And so that was kind of fun for a while, and I didn't have any help. I feel like academics, you have to be like this insane perfectionist, where at other places like government, maybe you do, and I should have probably put that higher. Sometimes in industry, you just say, okay, we got this, it's good enough, and I can't spend any more time on it, so let's just get to the next thing, right? Okay, so that's what I would think that it is. And this is the number one thing I would have to say when I talk to people in industry. They said that this is what they sort of liked the most, is that they weren't spent polishing every little sentence in a paper and getting everything exactly right, because you just didn't have time. Government probably has the least sort of outside of work. You need to work sort of time spent on it, although you still can. Like academics, I wasn't going to get any sleep most of the time, and it really depends on a startup. I think when we first started, it was equivalent to academics. It was like I am doing 6 jobs and I am never going to get it done, and it's going to be really hard. But now I have enough time, and since you know, startups are so great these days, I have my own startup because that's the way we roll. So with this, does anybody have any other questions? Do they have any other comparisons that they would like me to do? I didn't even factor in salary. Yes?
I have a question on the side gig. I know that in some companies you get a certain amount of time to work on your own projects.
This is not true here, no. In fact, if you're thinking about Google that did that, they did away with that. They did away with that a couple years ago, so that's not something they do. Google, so if you go with a big company like Google, so my PhD advisor, who was kind of an expert in machine learning, he was even at like Carnegie Mellon. He went to Google because he got sick of academics. He got sick of the pressure, and he went to Google. And at first, he started to do some things that were cool, like they had like the solar map where you could see like how much you would make if you could, how much electricity you could produce if you put so many solar panels on your roof, and it was live and it was streaming, and it would give you like the price per electricity that was going at where you were right then, right? It was like a very specific model, and they were doing like maps and doing like machine learning on maps and stuff like that. That project got canned, and he left because he was doing like writing JavaScript after 4 years. They took this person who was like sheer brilliant, and then they just kind of wasted it. So it's sometimes, again, you're kind of like forced to do something that you didn't really want to do. So now he's at an electronic medical records company, and gonna do machine learning again, and gonna try and make us healthier, is what he says. So, okay, other questions? Yes, please.
Well, because startups are hot right now, we've got another line to suggest for this table. How much of your outside work you get to own, and how much your employer is going to claim?
It depends on what it is. So it depends on how directly involved in what it is that you do as to what it is. So I was approached to do sort of some outside gigs that are directly in genomics. And so that I wasn't comfortable doing. That I wanted to sort of pull into the company and be able to do it through the company guys, like as kind of like consulting services for the company. Go ahead.
Did you hire a lawyer to read your contract before you joined the company?
I did not hire a lawyer.
Do you now know what your restrictions are?
It's been changed 3 times since I was there because of the lawyer who came on. And so the last thing, they were like less restrictive, actually. They were less restrictive. There was also a no-compete clause. Do we know what that is? That means that you can't do the same thing within a certain amount of distance for a certain time. And they had a no-compete clause, but they did away with that. Because really, you could say that, what's that?
They're unenforceable.
Exactly right. It's really unenforceable. It's almost even ridiculous, though. But when I did have this kind of idea for a startup, they were completely fine with it. And were somewhat supportive as long as it didn't interrupt what was going on. So I decided that I would like spend another chunk of time stressing myself out and trying to make like a physical product. I will show you what this startup is since we have a little bit of time. You can leave if you want, I don't mind. Base12tech.com. I swim a lot, and so but there's no internet connection.
Peter, you can get on, you should be able to get on CruzNet.
Oh, no, I'm
Oh,
sorry.
you're on.
That's
You're
okay.
on. Right. I swim a lot, and I swim for exercise, and I started to come up with crazy ideas. I call them like hypoxic thoughts. It's like the ultimate shower thought. It's like when you have low oxygen environment and you've been underwater for too long and it's cold and your brain is like kind of doing strange things. I kind of have strange ideas. My first strange idea was that I think the base 10 number system is terrible. That's like my first realization, and it's completely unnatural. So I think base 12 would be better. Other people have had this idea, and also I think it would catch on because we would call it the twesimal system instead. And that sounds so ridiculous and just so internet-y that it might actually catch on. I think we should be base 12, and we're not. yeah. That's all I'll say about that. So part of this is that, because I think it would also be easier in some ways to sort of digitize the world. I think we're coming, this is just me talking wildly now. I think we're coming to an era like every year, every day, our reality is more and more virtual. Like the virtual world and the physical world, the analog world, are kind of merging a little more. And I thought that I wanted to make it merge faster. So if you have a phone, right, if you have a Motorola phone, you can now snap on these devices like a projector or something like that, or a printer. You can actually snap on a physical printer. Where's my phone? I can't believe I don't have it, right? And so Motorola along with Indiegogo had a challenge called Transform the Smartphone, is to come up with a new version of a device. So somebody came up with one that's actually a mass spec. It's like can do like small molecule mass spec, like just on your phone. And so I thought, you know, I want to sort of merge the digital world faster. So I came up with this idea to create a snap-on that was called Orama. Let's watch my little pitch video. As it pitched it on Indiegogo and everything. Do we have sound? Why did I share it?
Martinez, founder and CEO of Base 12 Technologies.
Wow.
Today, we are proud to bring you our first offering, the Motorola Mod Orama. The Motorola Mod Orama is the first Motorola Mod that is a 3D augmented reality device. Snaps on just like any other Motorola Mod.
How awkward. How strange a person.
You can use it either as an augmented reality viewer, which can plug into any currently existing headset, or you can use it to capture 3D video. I was excited to first receive my Motorola Play and Z camera series. It was the perfect platform on which to create new experiences for a user. Orama's lenses are wider than the human eye, producing a unique sense of depth. So thank you for supporting our mod. We're excited to be able to bring you more products like Orama that enhance your connection to the world.
Okay, that was awkward. So that was my device, and I had like a series of devices, like one that would be like car integrated and sort of lots of other things. And so I think, yeah, it was, I did get the award. I did get the sort of grand prize finalist award from Motorola, which was a cash prize. So that was nice to see, and we're still under development, and I will still take it forward as far as I can. I actually wanted it to do it like, make it like a device for like sighted impaired or hearing impaired is like really where I wanted to take it for this particular thing. Not that somebody who can't see could see their phone, but hearing impaired might be able to. Anyway, so any questions about making your own startup? I can answer questions about making your own startup, joining somebody else's startup, doing all. Yes, please.
How about work-life balance?
Oh, work-life balance. Isn't that like kind of the most important one? Right. Well, if you torture yourself and make a second startup, you just threw your work-life balance away. That was a ridiculous move. But I would have, is it better? I would probably have to say government was probably the most free time. I would think that in industry, there are certain times where I am expected to be at work and it's like sort of a priority, but it's probably like, it's probably better than academics. It's gotta be, right? I don't know. I have no, it's hard to say. It's better, but yeah. Yeah, that's sort of, I would say it's sort of better, but you can make it worse no matter what. Right, and so if I were at the government, I wouldn't be able to do this technology thing. This would be like completely out of the question. There's no way I could do this around security. Okay. Okay, so that is it. I have kind of a slide on coping skills. Like make sure you take care of yourself, eat right, recognize your family structures. Don't be afraid to push back. If you think things are getting too hard, just push back. Say, I can't do that right now, leave, disappear. It's okay. I think when you're a postdoc, it's less so when you're a grad student, but postdoc, I think the most damaging thing is to isolate yourself. So try and join somebody. So like a good way is to like say, can I come to your journal club? Like try and interact with as many people as you can, because you can just sort of start working on your little thing and just get like completely walled off from the world and not realize what's going on. So definitely I think those things would have helped better. Right. Also anti-procrastination. So it's like, if you are a procrastinator and you feel anxiety when you look at something, which I definitely did and still do sometimes, it's like break it down into a small enough chunk that you can like accomplish that chunk, right? Let's do this one slide. Like let's make one slide and then we'll see how we feel. So break it down. If you're procrastinating, you're probably looking at things too big, too broad, and you're getting overwhelmed. So break it down into a smaller piece. And that is really, really helpful. Okay. Here's my kids. This is what keeps me going, I guess. They're very silly, and these are all the students and people who've worked for me. So you can still have students when you're in industries. In fact, we actually encourage it. Because it's cheap labor. Ha ha! And just kidding. I think that the one thing that the Ross Procrasta Genome Project taught me was that undergrads can be just as productive as other students. And so I'm glad to have anybody. All right, thank you very much. That's it.